Hello, everybody. My name is Maggie Beheler-Amass, and I'm here today to talk to you about dynamic gene regulatory network inference with interpretable biophysically motivated neural ODEs. Okay, so gene regulatory networks are complex networks of interactions that regulate spatial and temporal gene expression, and this allows organisms to respond to environmental, developmental, and physiological signals. These signals come into the cell and they're intercepted by proteins, and those proteins will send messages to transcription factor proteins, which will go on to bind to DNA to regulate gene expression. And the overall goal of transcriptional gene regulatory network inference is to uncover which transcription factors are regulating which genes from a systems level perspective and represent this as a network. And once we have our underlying gene regulatory network, we can use it as a model to predict outcomes of experimental perturbations. So let's say we experimentally perturb our system with a disease or a drug. We can see how these perturbations change regulation and therefore change the connections in our gene regulatory network. So another way to think about this is starting our system in some initial state and then observing a phenotypic response to a perturbation over time. And so overall, we want to build models that are causal, where we can say that this perturbation in a gene regulatory network causes this observed response so that we can perform predictive perturbations in silico that uncover the chain of events in our biological system. And so this leads us to dynamic gene regulatory network inference, where we'd like to build these predictive models. And so the first goal of these predictive models is to be able to build a causal model that takes in some dynamic data and like some dynamic gene expression and uncover the dynamical system that's driving the phenotypic response that we observe. And so this is our gene regulatory network. Next, we'd also like to be able to predict future states of the system. So we can train our model on a cell responding to a perturbation. Let's say that we add a drug to our data set. We'd like to be able to predict the future response to that perturbation at unseen time points. And finally, we'd like to be able to predict responses when we start our system out at some new initial state. So let's say that our new initial state is a disease cell, and we add the same drug that we used in our previous example that we had applied to healthy cells. Can we predict how this disease cell will respond to the same drug over time, starting out in this new initial state? And if we can achieve all of this from one model, we will be able to predict, potentially predict experimental outcomes in silico, and this could create a really powerful tool moving forward. And so one candidate biophysical model that we can use to do all of this is a linear differential equation that models the change in expression over time, or in RNA velocity, as a function of mRNA degradation and transcription. And so here, lambda is mRNA half-life, and that is applied to gene expression, it gives us an mRNA degradation, and that's the negative component of RNA velocity because the mRNA is degrading. And then alpha here is transcription, which is the positive component of RNA velocity because the mRNA is being produced. And so we're trying to infer and learn these different positive and negative components of RNA velocity out of this model. And so one way that we can do this is, and we can use this biophysical model with a deep learning technique called neural ordinary differential equations. And so the overall goal of these neural ODEs is to find a differential equation f using a neural network to parameterize or describe a vector field dx dt based on some input data x. And so in my case, our learned vector field is going to be RNA velocity, and it's based on some input gene expression. And so the cool thing about neural ODEs is that once this vector field is learned, it's a continuous function that can be evaluated anywhere in time. And so if we know the RNA velocity anywhere in time, we can use it to predict gene expression at any future time point. And then by feeding this estimated RNA velocity into our ODE solver, this acts in the same manner as integrating over our function f that we've learned from some initial time point to a future time point. And we can add this to our starting time point in a continuous manner, and then we'll be able to predict future gene expression at some time point n. And then by minimizing the loss between the actual gene expression and the predicted gene expression at this future time point n, we can train our model. And so to put all of these pieces of this biophysical model together, we can use the neural ordinary differential equation to learn a predictive model for transcriptional regulation while implicitly learning these biophysical parameters. And so this model estimates an RNA velocity as a function of mRNA degradation and gene expression and transcription. And this is all approximated by the neural network trained solely on gene expression. And so we can implicitly learn RNA velocity, mRNA degradation, and transcription directly from the model. And we can use these estimated values to infer our dynamic gene regulatory network. So bringing back this idea of causal modeling, if we just use a deep learning framework, we could correlate changes in gene expression with each other, but this isn't really learning a causal transcription factor regulating which genes gene regulatory network. And so we'd like to be able to interpret our model to know precisely which transcription factors are actually regulating which genes. And so the interpretable deep learning architecture for gene regulatory network inference that I'm using is based on an autoencoder model that I worked on previously and we developed in our lab. It was published in Genome Biology this year. And the way that this model works is that gene expression is embedded into the latent layer, which is identifiable as transcription factors due to a knowledge constraint mask in the form of an adjacency matrix of known regulatory interactions between genes and transcription factors. So we're pruning away connections between these genes and transcription factors based solely on known regulatory interactions in this knowledge constraint mask. And then the gene regulatory network, or the connections between the transcription factors and their target genes, can be identified by removing individual transcription factor nodes post-training and measuring how much the change in error in the model prediction changes with and without this transcription factor. So this is the original, this blue here is like the original R squared for this gene. When we remove this node here, we see that the predictive capability of this model for this gene decreases. And so we can do this across all transcription factors and use this to rank the importance of how much a feature or this transcription factor is regulating this gene. And then finally, we can use this ranking to uncover which transcription factors are regulating which genes, which is our gene regulatory network. So putting all of the pieces of this model together, my model takes in expression at an initial time point ti, and it feeds it into two neural network blocks, one representing the half-life of mRNA lambda and one representing transcription alpha. And so the transcriptional model has this interpretable deep learning architecture that infers the gene regulatory network of transcription factors regulating the genes. And then the outputs of each of these networks are constrained to be positive using the ReLU activation function so that we can directly interpret the outputs of these models as biophysical parameter estimates. A negative sign is applied to lambda to disentangle this negative aspect of RNA velocity from the positive aspect of RNA velocity. And then we combine the outputs of these two models to get our RNA velocity estimate at a certain time point. And then we use this ODE solver to integrate over our RNA velocity and estimate expression at a future time point. And I use mean squared error to train the model to see how accurately the model can predict expression at a future time point versus the actual expression. So the model system that I'm using for this project is yeast responding to a drug perturbation. That perturbation is rapamycin, which affects the TOR complex, and it creates a starvation-like response in yeast. And this induces an upregulation of stress response and metabolic transcripts and a downregulation of ribosomal transcripts. So this was created by a postdoctoral researcher, Chris Jackson, in my lab. The data is available here. It's a really amazing data set. And basically what he did was he grew yeast in rich media for 20 minutes, and he continuously sampled that these yeast using a single cell sampler. And then at 20 minutes of growing these yeast, he then added rapamycin and then continued to continuously sample the data for another 60 minutes. And so we get this really nice continuous time axis of these single cells responding to a drug. And so in this plot here, it's a UMAP where each of these dots is a single cell, and the colors represent the time pool that they were sampled from. So these are in the first 20 minutes, and then we see this nice time axis response. And so we can see it's a really continuous data set of these yeast responding to the drug perturbation. And it's also a very well-known system, so we can use it to test our dynamic gene regulatory network inference methods. So this is some validation data here. This is RPL3, which is a ribosomal protein gene, and it turns off in response to rapamycin. And then GAP1 is a stress response gene that's turning on in response to rapamycin. So I split the data into 80% for training and 20% for validation. So this is all validation here. And I arranged the cells into trajectories of 40 minutes at sampling these cells 1 minute apart. But if we look at this data set, let's say this is my initial expression at time 0, then if I go to time at the expression at minute 1, I could make a trajectory that goes from up here to down here and then up and down. And that is actually, it was really challenging for the neural ODE to learn on. And so what I did to overcome this challenge was I took the mean of 20 trajectories to try to give the model a chance to learn on a smoother data set. And this proved to be quite useful for the model. But the way that this works is that I gave the model an initial cell state from this validation data at time 0, and then I asked it to predict the next 40 minutes of expression. And when I do that in red here, you can see that the model is able to predict these trajectories along this validation data very, very well, which is really exciting. And just for a little like dimensionality idea, this data set is around 5,700 genes with around 200 transcription factors. And so it's predicting these gene expression profiles well, which is exciting. Then I also wanted to see how well this model could predict RNA velocity. And due to the continuous nature of the data set, we are able to compute an instantaneous velocity per cell based on local neighborhoods of time-ordered cells. So we could look at this cell here that's at 17.4 minutes and look at the expression in that cell. And then we could look at this cell at 34 minutes and say, how much is this gene expression changing over time, just using this little linear estimate. But we can use that linear estimate to then compare against this implicitly learned RNA velocity that's getting learned from the model. And again, the model is doing a really great job, which is really exciting because the velocity isn't provided to this model at all. The model's being trained only on expression data, but it is learning implicitly this velocity with really high accuracy. So finally, I trained the model on 40 minutes, and I wanted to then see how well it could predict 20 minutes of withheld data to see how well it can perform at these future predictions at unseen time points that I talked about earlier in the talk. And so for RPL3, the model is predicting the future state of the system quite well. And for GAP1, it's doing some good predictive performance, but it's failing to see that GAP1's going to decrease in expression over time. But that might be due to the fact that we're predicting the future state of the system. And so for the model, it's assuming that there's no change in regulation. It's just continuing this program that it saw in the data that it was trained on. And so the fact that it's missing this prediction here means that the gene program that's running has probably changed. There's some alteration in this data that wasn't trained on, so the model can't predict it. So in this case, it might be the fact that GAP1 is saturating in these cells and then it's being repressed as it saturates. So just to give an idea of an overall predictive performance, I trained the model across 5 cross-validations, sampling the data differently each time. And as you can see, the model is really, really stable and has a really high predictive capability across all 5 of these iterations, looking at this 40-minute trajectory and then also at the 60-minute trajectory where it's asked to predict data that it didn't see in between 40 and 60 minutes. And then also I wanted to see how well this model is actually performing gene regulatory network inference. And so the way that I do this, going back to this knowledge constraint mask, which is these known regulatory interactions between genes and transcription factors in the form of an adjacency matrix, I split that adjacency matrix where I withheld 20% of these known regulatory interactions during the training of the model. And then I measured the AUPR, which is how many of these withheld interactions are being learned from this inferred gene regulatory network. And so the value is pretty stable around 0.07, which is low for AUPR, but an AUPR was also calculated for this data set when this data set was originally published. And my collaborator was getting around 0.1 for his AUPR. And so we still find this result to be pretty promising. And we think that the reason that one of the reasons that the AUPR might be so low is that this is just trying to capture a gold standard of interactions between genes and transcription factors in yeast on a data set that's only responding to this drug perturbation and rapamycin. So it might not be able to recover all of these rich media interactions that you'd normally see. So overall, neural ODEs provide an interpretable architecture for biophysical models, and incorporating these biophysical elements increases a systems level understanding of regulation. The model can accurately infer dynamic regulation, and it predicts expression in silico, and the model can implicitly learn RNA velocity directly from the expression data. So future directions, I would like to apply this model to larger data sets, like potentially a hematopoietic data set. And I also need to continue improving the other biophysical estimates that are being implicitly learned in the model, like the RNA half-life and also the transcriptional output. So thank you so much to my collaborators, Chris and Andreas and Rich. They've been a great help in this project. Thanks.
Thanks for the nice presentation. I was wondering, the selection of the input data, so the time points you need to train a model seems like crucial. Did you try leaving out time points and see what is kind of minimal information you need to get to a good model?
Yeah, I tried also like giving the model before the drug was added. So there's like minus 10 minutes before, just to see if it could learn when genes were turned off, if you could see them start to turn on. And then I also looked at how long the model could predict accurately for. So it seems around 40 minutes is the optimal length of a trajectory. When it goes to 60, it starts to over and underfit a little bit. But I haven't tried leaving out middle sections, which I think could be interesting to see if you give it the starting and the ending, can it discover what's going on in between there as well.
Thanks.
Yeah.
Hi, thanks for a great talk. So I wonder, did you compare with like SC-Velo to see if your velocities are, comparable with their estimates?
I didn't do that, but in the paper that the data set is originally published in, my postdoc that I work with did a giant comparison against velocities and different pseudo-time trajectory creations. I think I should do some benchmarking against SC-Velo and see how it compares.
Yeah, that would be great. And another question, when you do this in silico knockout, do you look at the change in velocity or gene expression?
I have not looked at that yet, but in this bioRxiv paper, there is some perturbations of those transcription factors, and you do see a change in expression. Definitely, yeah. So that in silico perturbation is working.
Hi, amazing talk. So yeah, the previous person also mentioned about using pseudotime instead of time series. So in the case of pseudotime, you might be able to get, because in your training data, I suppose it's more of like a linear change of expression from one condition to another. And in terms of pseudotime of cells can have more complex structures like tree-like structures. What do you think your model have do you think your model has the potential to accommodate more complex changes of the cell states?
I really hope so. I think it would be very interesting to see, right, if with a hematopoietic data set, if you can get different bifurcation points, right?
Yes,
yeah,
it would be very curious to see how to explain the bifurcation using the dynamic GRN.
And I think that neural ODEs are well suited for that, right? Because that's part of their like oscillatory functions and things like that.
Thank you so much.
Hi, thank you very much for this brilliant talk. It was really good work. I was wondering, do you think you might be able to account for this error that you have when you go towards predictions that are a bit further away, like 60 minutes? Do you think you could account for that if you include cell cycle information that you could train from before?
Yeah, there is a cell cycle component of this data set as well that I thought I could have ready for this talk, but not quite. But I do think it would be very cool to try to jointly train on those and then try to combine the information later to see if they can learn together.
Cool.
Yeah.
Do you have already some preliminary data or results for that?
I do,
Yeah.
which are on my laptop if you'd like to see them.
Oh,
Yeah,
absolutely.
Okay.
Would be
yeah.
keen. Thank you very much.
That'd be great. Thanks.